My dear students, good afternoon. Today I will deliver the lecture on bioremediation of problematic soils through uh, through multi-purpose tea species. This comes under the course of problematic soils and their management. It is from the unit number 4.1. Uh, I will cover this whole lecture in the different outlines such as objective, introduction, definition of bioremediations, types of bioremediation, technique of bioremediation, bioremediation of problematic soils through multipurpose tea species, conclusion, learning outcome and references. The objective of this today lecture is to introduce the students about bioremediation of problematic soils through multipurpose tea species. You know, in agriculture practices for crop production, many harmful chemicals are used to control insect pest diseases and weeds and applied to plant as well as to soil. The chemicals are, however, normally detoxified in soil in a reasonable time if used in recommended amount. The detoxification of such chemicals done by soil organisms. Bioremediation covers various types of contamination of soil and diverse technique of their treatment. However, truly speaking, bioremediation involves the use of microorganisms in association with plant soil amendments and agronomical practices to remove counted or render a harmless effect of the soil contaminant. Sometimes instead of bioremediation, the term phytoremediation is also used when contaminated soils are exposed by different kind of vegetation. You can define the bioremediation like this. Bioremediation is a branch of biotechnology that employ the use of living organisms like microbes and bacteria in the removal of contaminant pollutants and toxins from soil, water and other environment. This bioremediation may be used to clean up the contaminated groundwater or environmental problems such as oil spills. Truly speaking, the bioremediation is a worldly meaning is bio means jav and remediation means sudhar. There are different types of bioremediation based on the place where water waste are removed. There are principally two ways of bioremediation. One is in situ bioremediation. Most often in situ bioremediation is applied in eliminate to pollutants and contaminated soil and groundwater. It is a superior method for the cleaning of contaminated environments because it saves transportation costs and uses harmless microorganisms to eliminate the chemicals contamination. Two types of in situ bioremediation are distinguished based on the origin of the microorganism applied as bioremediations. Intrinsic bioremediation. This type of bioremediation is always done in situ and it is carried out without direct microbial amendment and through intermediate in ecology condition of the contaminated regions and the fortification of the natural population and the metabolic activities of indigenous or naturally existing multi microfauna by improving this nutritional and ventilation condition. Engineered in situ bioremediation. This type of bioremediation is performed through the introduction of certain microorganisms to a contaminated site. As the condition of contaminated site are most often unfavorable for the establishment and the biodiversity of the exogenous amended micronutrients. Therefore, here like intrinsic bioremediation, the environment is modified and away so that improved physico-chemical condition are provided, oxygen, electron acceptors and the nutrient, for example nitrogen and phosphorus, are required to enhance the microbial activity. In situ bioremediation, this process of bioremediation here takes some place somewhere out the contamination site and therefore requires transportation of contaminated soil, pumping of groundwater to the site of bioremediation. This technique has more disadvantage than advantage. Depending on the state of the contaminant in this step of bioremediation, ex situ bioremediation is classified as solid phase system including land treatment and soil piles. The system issued in order to bioremediate organic waste and problematic domestic and industrial waste, sewage sludge and municipal solid waste. Solid phase soil bioremediation including three processes: land farming, soil biopiling and composting. Slurry phase system including solid liquid suspension to bioremediator. Slurry phases bioremediation is a redirectly more rapid process in compared to the other treatment process. This is the bioremediation is a triple corner process. So mostly the pollutants, they are of two nature: inorganic and organic. Inorganic, there is solid phase, liquid phase and gas phase, and in this environment, the environmental, the soil, water and air, and in organisms is the microorganisms, plants and enzymes. They work is together in the bioremediation of triple courses process. Bioremediation techniques. There are several bioremediation techniques. Some of them have been listed as such: bioaugmentation, the addition of bacterial culture to a contaminated medium, and frequently in situ process to factors limit the use of added microbial culture in a land treatment unit, or non-indigenous cultures rarely complete well enough with an indigenous this population to develop and sustain useful population levels, and be most soil will long term exposed to biodegradable waste have indigenous microorganisms that are effect degrade if the land treatment unit is well managed. Biofilter, the use of microbial stripping columns used to treat air emission. Bioreactor, the use of biodegradable processes in contaminated area or reactor for the biological treatment of relatively small amount of waste. This method is used in used to treat slurries or the liquid slurries. Reactors or aqueous reactors are used for ex situ treatment of the contaminated soil and water pumped up from a contaminated plumb. Bioremediation in reactors involves the processing of contaminated solid soil sediments, sludges, or water through an engage, engineered contaminated system. A slurry bioreactor may be defined as a contaminant vessels and the apparatus used to create a three-phase solid, liquid, and gas mixing conditions to increase the bioremediation rate of the soil-bound and water-soluble pollutants. As a water slurry of the uh, contaminated soil and biomass, usually indigenous microorganisms capable of degrading target contaminant. Fourth is biostimulator, the stimulation of the indigenous microbial population in soil and the groundwater. This process may be done either in situ or ex situ. Bioventing is the process of the drawing oxygen through the contaminated mediums to stimulate microbial growth and activity. Bioventing is the most common in situ treatment and involve supplying air and these nutrients through well to contaminated soils are similar and stimulate the indigenous bacteria. Bioventing involve low airflow rates and provide only the amount of oxygen necessary for the biodegradation while minimize volatilization and release of contaminants to the uh, atmosphere. Next is composting, an aerobic and thermophilic process that is mixing contaminated soils and a bulking agent. Composting may be preferred using static piles. Aerial piles are the contaminant fed reactors. Composting is a technique that involves combining contaminated soil with non-hazardous organic amendment uh, such as manure or agriculture waste. The presence of these organic materials supported the development of a rich microbial population and elevated temporary temperature characteristics of the composting. Next is land farming and land treatment prepared with bioreactors. Solid phase treatment system for contaminated soils may be applied as an in situ in process of ex situ in a soil treatment cell. Land farming is a simple bioremediation techniques in which contaminated soil is excreted and spread over a prepared bed and periodically tilled until pollutants are degraded. The goal is to stimulate indigenous biodegradation microorganisms and facilitate their aerobic degradation of contaminants. In general, the practice is limited to the treatment of specific 10 to 35 centimeters of soil. Biopiling. Biopiling are a hybrid of land farming and decomposting. Essentially, engineering shells are constructed as aerated composted piles, adding compost to contaminated soil enhanced by remediation because of this structure of the organic compost manure mixing. Another is the phytoremediation. The phytoremediation deals with the cleaning up of organic pollutants and heavy metals contaminated using plants and rhizosphere microorganisms. It is a inexpensive, eco-friendly, and of efficient means of the restoration of the polluted environment, especially those of the heavy metals. Nevertheless, the level of soil contamination, the quality of metal contaminant in the soil, as well as the ability of plants to aggressively take up metals from the soil determine the success of phytoremediation at any polluted site. Plants utilized in phytoremediation are the hyper accumulators with the very high heavy metal accumulation potential and little this biomass efficiency, and non-hyper accumulators which possess lesser extraction capacity than hyper accumulation, but whose total biomass yield is substantial higher and are fast growing species. This is the process involved in the phytoremediation. It is shown. It is already shown in this figure. Phytoremediation, this volatilizations, phyto extraction, and metal chelation, and this uh, bioremediation of the heavy metals. Advantage of bioremediation: first is bioremediation is a natural process and is therefore perceived by the public. Bioremediation is useful for the complete destruction of a wide variety of contaminants. Instead of transferring contaminants from one environment medium to another, for example, from land to water or air, the complete destruction of this target pollutants is possible. Bicarbonate can often be carried cut on site, often without causing major disruption of the normal activities. Bioremediation can prove less expensive than other techniques that are used for the cleaning up of hazardous waste. These are the disadvantages of bioremediation. Bioremediation is limited to those compounds that are biodegradable. Not all compounds are susceptible to rapid and complete degradation. There are some concern that the products of the biodegradation may be more persistent or toxic than the parental compound. Biological processes are often likely specific to the microbial population, suitable environments, growth conditions, and the appropriate levels of the nutrients and contamination. It is difficult to extrapolate, deduce from the bench and piled or scaled studies to full-scale field operations. Bioremediation often takes longer than the other treatments options. Bioremediation of problem soil. Remediation means to solve a problems, and bioremediation means to use biological organism agent to solve an environmental problems such as contaminated problem soils or contaminated groundwater or biodegradation in the use of microbes to clean up the contaminated soil and groundwater. Microbes are very small organisms such as bacteria that lives naturally in the environment. Bioremediation stimulates the growth of certain microbes that use contaminants as a source of food and energy. Contaminants treated using bioremediation include oil and other petroleum products, solvents, and the pesticide. Problematic soils are those soils which are not suitable for the arable farming because of specific limitation In general, problematic soils are two types. One is physical problem soils and another chemical problematic soils. Agroforestry system have this potential tools to make use the marginal and the degraded land through the soil improving effect of the trees. It proves to be one of the cheapest and best methods for the reclamation of all the problematic soils. Agroforestry system like silviculture, silviposture can improve the physical and chemical property of the soil along with the additional return on long-term basis. Bioremediation of the physical problem of soil. For the bioremediation of the physical problem of the soil, like sandy soils, subsoil, hardening or hard pans, surface crusting, waterlogged soils, peat and marshy soils, etc. Tree species like uh, eucalyptus, robusta, swamp, mahogany, and Syzygium cuminatus and jamun, Terminaria Arjun, Selex, Tetrasperma, Indian willow, and Dalbergia latifolia sesame, Eucalyptus, Camendula river red gum, Eucalyptus grandis, rose gum, and some grasses like this back, Baccharia, Multifolia, paragrass, and the cyanodactylon, dew grass, Bermuda grass, Dactylon, Cassicula, and Bracilla decumbena, etc. are the commonly used for the bioremediation or by the bioremediation of the problematic soils. Bioremediation of chemical problem of soils like different chemical problems of soils, saline, sodic and saline sodic DC also reclaimed or managed by the following tree and grasses respectively. The saline soils are the promising woody species, the Salvadora persistica, mustard tree, Prosophis julifera, mosquito tree, Acacia nautica, babul, Buteria monosperma, Palash, Termendaria, Arjun, Salexis species, Indian willow, Delvergia shisu, Casuarina, Costifolia, Sharu. Heavy salt tolerance and high biomass producing grasses species include this Europus langipodis, mangrove grass, or rabbit foot, Eleuropus, and then Sedia futicosa, Serbi, sea blight, and cyanodactylon, Bermuda grass, doob grass, and branches ramra, brown top millet. Sodic soils, mostly Prosophis julifera, mosquito tree, and Disothium analutum, canal grass, improve the soil conditions to such an extent that after some time or years, some fodder species can be grown under trees such as Berseem, Typholium exotica, Segi, Melitutus, this Provifula, and Shaftal, Typholium resu pinnatum. There is a relationship tolerance of the fruit trees in sodicity. Mostly the exchangeable sodium percentage ESP for sodicity is given. If this ESP exchangeable sodium percent of any soil is below 20, only sensitive plants can be grown like Mangifera indica, mango, Heterocarpus, Heterophilus, jackfruit, Musca, Paradisca, banana, and low 20 to 30 this exchangeable sodium percentage, sodium guava, citrus lemon, and vitis vinifera grapes. Medium 30 to 40 ESP, the Panica canistura pomegranate, and at high exchangeable sodium percentage, this can be Gigi-Phus jujuba, Bayer, Tremendous indica, Termerend, and Manicera jubata sapota, Limna acidissima, wood apple, and Phenotix dexti fer date palm. Saline soil soils, these plants can be very useful. This Acacia artiliformis, the Akashmoni, Ajardica indica, Neem, Casuarina equisifera, Ceru, Delvegia Shiso, Elanthus excels, Tea of Heaven, and Prospis serifera, Khejri, Acacia toleris Israel bubble, and Acacia notifolia bubble. Now we can conclude this whole lecture that bioremediation involves microorganism soil amendments, multi-purpose plant species for bioremediation of problematic soil, which help to remove the contaminants of the soil. Besides bioremediation, phytoremediation is also very important, such as cellular algae, some of the hydroponics like this jalcumbi, water hyacinth, and canare may also be used for the remedial of water and air pollution. The details of the bioremediation process, like techniques, methods, and use of multipurpose tea already mentioned as above, will be quite useful for the bioremediation of physical and chemically degraded problem soils. The learning outcome of this lecture is, in the last students are quite competent to know the bioremediation, its type and techniques of bio and phytoremediation of problematic soils through using various multipurpose tea species in this contamination soil and water and air. I have taken the materials from different references which have cited here with the Rishikesh Thakur and Tell multipurpose tea species for bioremediation of problematic soil, Fundamental of Soil Science 2011, IARI publication, New Delhi, Abhishek PC, Srivastava S, and Singh, comparative bioremediation potential of 4 rhizosphere microbial species against Indian and vertebrae, this phytoremediation as a management option for the contaminated sediments, and the Vert V. Ital, this metal immobilization and soil amendments efficiency of a contextual sediments landfill site etc. So all the matter is taken from these references, and I have already given this matter as a PPT and a video. You please go through the video and PPT and come with prepare in the this webinar class, and if there is any query you can ask by the raising the question. Thank you very much.